Yeah.
Do you see like the
She
advancement of
sees,
slides?
you see the big slide, right? Yeah, okay. She's good.
And let me bring up my pointer. Okay, well, we can start now after all these technical difficulties. So thank you, Dr. Greer, for your kind introduction, and hello, everyone, and welcome to my seminar. Today, I'm going to talk about investigation of Factor VIII gene therapy, enhancement and persistence of Factor VIII transgene. So to start, I'm going to briefly talk about Factor VIII biology, hemophilia A, its current treatment approaches, viral gene therapy, recombinant, focusing on recombinant AAV gene therapy. Then I will proceed with presenting my projects, hypotheses, the results that I've got so far, and the future steps that's remaining for each project. So Factor VIII is a coagulation protein encoded by Factor VIII gene. Factor VIII gene is a very large gene consisting of 26 exons located on chromosome X. Factor VIII is mainly synthesized in specific endothelial cells in liver called liver sinusoidal endothelial cells, and to some extent in extrahepatic endotheliums like lymphatic and glomerular endothelium. Factor VIII gene encodes this large glycoprotein of Factor VIII, which has 6 discrete domains. And then it is folded in ER-Golgi apparatus and then released to the circulation in an inactive form in complex with 1 Willebrand factor or VWF. In the coagulation cascade, once Factor VIII is activated, the VWF is dissociating from Factor VIII, and now Factor VIII can act as a cofactor to help Factor IXA in cleavage and activation of Factor X, which eventually results in the continuous of coagulation cascade and fibrin clot formation. So it has a very important role in 10-ACE complex of coagulation cascade. A little bit about hemophilia A. Hemophilia A is an X-linked recessive bleeding disorder affecting 1 in 5,000 male births each year. It is caused by pathogenic variations in Factor VIII gene. About 45% of these variations are intron 22 inversions. Other variations reported in the literature are large and small deletions and insertions, missense and nonsense variations. These variations, how they affect Factor VIII is that they result in the reduction of Factor VIII activity in the plasma. And based on this reduction, the disease is categorized into 3 types of severe, moderate, and mild hemophilia. Please note that normal Factor VIII activity levels in plasma is 50 to 150%, and severe cases of hemophilia, which comprise like maybe 50% of the population of hemophiliacs, have lower than 1% plasma level Factor VIII activities. In severe cases on hemophilia, we see recurrent spontaneous bleeds into joints and muscles, and if left untreated, they will develop severe arthropathy. Currently, there are 2 types of treatment approaches used for hemophilia A. First one is coagulation factor replacement therapy, in which patients receive plasma-derived or recombinant Factor VIII every 2 or 3 days. Or in cases of bioengineered recombinant coagulation factors, they can receive the factor every week. The major complication with this replacement therapy is the development of anti-drug antibodies termed as inhibitors, which renders the treatment ineffective in about 30% of the cases. The alternative approach, which gained popularity, is emicizumab, which is a bispecific monoclonal antibody which mimics the activity of Factor VIII. The advantage of emicizumab is that it can be administered subcutaneously and biweekly, or in some cases monthly. So it really improves the quality of life of hemophiliacs. The other treatment approach is gene therapy, which actually offers cure for patients by providing endogenous expression of Factor VIII. And due to its relevance to my project, I will be focusing my talk on gene therapy using viral vectors. In viral gene therapy, the transgene of interest is introduced into a virus and then infused into patients in an in vivo or ex vivo manner. In the case of hemophilia A, we do that in an in vivo manner. There are various types of viral vectors that are used in viral gene therapy. And in hemophilia A, AAVs or adeno-associated viruses have been selected because of their low immunogenicity and the fact that 95% of AAVs persist in the nucleus as non-integrating episomes. So a little more about AAVs. Adeno-associated viruses was first described in 1965 as a contaminant of adenoviral stock, virus stock. In the figure here, you can see these small dots that are actually AAVs, and this bigger dot is adenovirus. And if we compare the size, we can see it is at least 1/5 of an adenovirus. So we can see that it is a small virus with small vector genome, a virus genome. It is a replication defective virus, which means that it needs help from a helper virus for its replication. It has a single-stranded genome, which is packaged into a capsid protein. It is considered to be non-pathogenic in humans, but early in childhood, about 50% of population encounter AAV infections. So there is the chance of humoral immunity against vector virus capsid in 50% of the population. So on the left, we see a genome, a viral genome of a wild-type AAV. We can see that it has a pretty simple structure consisting of 3 main parts, the rep and cap gene responsible for replication and capsid formation, and these 2 ITRs flanking the rep and cap gene, which are integral for virus replication and DNA encapsulation. The viral genome is then packaged into an AAV capsid, which can contain no more than 4.9 kb of vector genome. So a very limited packaging capacity. In order to make a recombinant AAV, almost all parts of the vector, the viral genome is removed and replaced with a transgene of interest and some other regulatory elements like enhancers and a tissue-specific promoter. In the case of hemophilia A, the promoter that we use is liver-specific, as hemophilia A is a liver-directed gene delivery approach. And the transgene that we use is obviously Factor VIII, which is B-domain deleted to reduce the size of Factor VIII cDNA from 7 kb to 4.5 kb, so that we would be able to incorporate that cDNA into a small capsid. So as we can see here, there is actually not much enough room for us to use regulatory elements. So we are very limited by the size of the AAV capsid, and the regulatory elements that we can employ within this expression cassette should be less than 400 base pairs. So it is very important to identify enhancers or regulatory elements that has high transcriptional activity and are small in size. Let's have a quick look at AAV transduction and AAV trafficking. AAV enters the cells through cell surface receptors, then it's internalized in endosomes. Upon endosome acidification, the vector escapes and enters the nucleus, where the capsid uncodes and synthesis of the second strand occurs. As I mentioned earlier, 95% of the vector persists in the nucleus as this episomal non-integrating circular forms, which give rise to transgene expression, protein synthesis, and then secretion into the plasma. my project and my research focuses on the factors that affect steps 6 and 7 of this AAV journey, and we try to investigate the factors that can affect vector persistence and transgene expression. Currently, an important limitation of hemophilia A gene therapy, first is the variability that we see between different patients and the decline in transgene expression. In the figure, you can see a result of clinical study in 17 hemophilia A patients, which received AAV5 vector. As we can see, a vector increase of expression in about 25 to 35 weeks post-vector delivery, but then we see a decline in the transgene levels, which remains till the end of the study. And actually, this decline that we see in transgene expression is one of the major disincentives for hemophilia A patients to try to be able to and to be willing to choose gene therapy for their treatment. What causes this decline is still unknown, but there are some reasons that we are willing to work on. One of them would be progressive loss of vector, reduced Factor VIII mRNA production, insufficient transgenic protein trafficking and secretion. So with this background, I'm going to propose the projects that I've been working on. The first project is to investigate the efficacy and safety of a novel enhancer element derived from the native Factor VIII locus for application in AAV-mediated Factor VIII gene therapy. Project 2 is to evaluate the effect of gut microbiome on the AAV-mediated gene delivery. And in Project 3, which kind of binds projects 1 and 2, is to investigate potential molecular mechanisms responsible for the decline that we see in Factor VIII expression. So starting with Project 1, we hypothesized that the enhancer element derived from the native Factor VIII locus will provide enhanced and more durable transgene expression. Factor VIII Padua is a hyperactive form of Factor VIII caused by a large rearrangement in Factor VIII gene. In the figure, you can see Factor VIII, which is transcribed from right to left. Factor VIII Padua was first identified by Simeone et al. In 2021 in 2 Italian families which manifested early-age thromboembolism and had persistently high plasma levels of Factor VIII, near 400% of Factor VIII activity. So they did a genetic analysis in these groups of individuals and identified a large duplication in the beginning of Factor VIII gene, which encompassed promoter, exon 1, and a large part of intron 1. They further investigated these regions to identify transcriptionally active marks within these regions, and they introduced different regions that were thought to be a putative enhancer elements. So we got interested in this duplication, and one of the regions within this duplication was fragment C, which you can see here. This is an in silico analysis of fragment C that has 927 base pair, and it has marks of DNA signal, which is a mark that suggests active transcription. Also, it is enriched with TF binding sites identified by Jasper. So we selected this enhancer element for our study. Also, keeping in mind that eventually we are going to use these enhancers in an AAV vector, we considered we needed a shorter enhancer. So within this fragment C, we also selected a smaller part with 400 base pair, and we call it fragment C prime. And we selected this region because of the peaks of TF binding sites and because it was more conserved among species. So these two enhancer fragments were cloned in a luciferase reporter plasmid upstream of a minimal promoter in both forward and reverse orientation. And then it was several cell lines were transfected with these two plasmids containing enhancers. One of the cell lines was hepatocytes and human lymphatic endothelial cells. As we can see in the figures, all enhancer elements were able to increase luciferase activity in these two cell lines. However, it was the fragment C prime, or the shorter enhancer element, that gave rise to a more significant luciferase expression. Same results and trends were observed in HEC293 and HUVEX. In order to complete our in vitro studies, we also introduced these enhancer plasmids in our C57 black 6 mice, where these mice received these plasmids through hydrodynamic delivery. 2 days post plasmid delivery, we performed bioluminescence imaging to check for luciferase expression, and 5 days post delivery, we euthanized mice to collect liver in order to measure luciferase in liver homogenates. In the figures, you can see study mice that were dosed with plasmids containing enhancer elements. This is a mice that received empty vector, just the vector backbone, and we can see no expression in these mice. But we can clearly see luciferase activity in mice that received plasmids containing enhancer. And this is the luciferase results from liver lysates, which confirm our in vivo imaging. So what we are trying to achieve right now, again, we are restricted by size. So we want to, again, reduce the size of enhancer fragments. In our results, we saw that fragment C prime, or the 400 base pair enhancer fragment, was to be more potent in increasing luciferase activity. So what we are trying to do is to select different regions of these enhancer fragments, these 3 regions spanning from 100 base pair to 200 base pair, and we will redo the same experiments, in vitro and in vivo experiments, with these smaller fragments, just to confirm their enhancer potential. And once we do that, we will proceed to the development of AAV vector containing one of these potent enhancer elements that is less than 200 base pair. So on the left, you can see a proposed expression cassette, which contains murine factor VIII cDNA, a liver-specific promoter, and this novel enhancer that we tested its efficacy. As a control, we will be developing another AAV vector. Structurally, it's the same with our study vector, and the only difference is that in here we will use standard transthyretin liver-specific enhancer elements, just to be able to compare these two enhancer elements. The AAV vector will be introduced into hemophilic mice. Mice will be followed for 1 year, and within 1 year, we will be collecting blood in a biweekly manner. And we also will have 3 different endpoints, in which in each endpoint, a group of mice will be euthanized and the liver will be collected. The livers that we collect will be used in Project 3 that I will elaborate on later. The plasma samples that we received during this 1 year will be used to evaluate Factor VIII activity levels using chromogenic assay. Moving on to Project 2, we are going to evaluate the effect of gut microbiome on AAV-mediated gene delivery. As I mentioned earlier, hemophilia A gene delivery is a liver-directed modality. And in this study, for the first time, we are investigating the effects of inflammatory liver environments that's induced by administration of antibiotics on transgene protein expression. A little bit of background on gut microbiota. The human gastrointestinal tract contains abundant and diverse microbial community that gathers more than 100 trillion microorganisms, including bacteria, fungi, viruses, which make up the gut microbiota. Each healthy human has a unique gut microbiome, which is formed in early childhood. The intestinal microbiota actually has this symbiotic relation with the host, and it's integral for many physiologic processes, including but not limited to food digestion, vitamin synthesis, immune system, and inflammation. A dysbiosis is a condition where there is an imbalance in the microbial composition, and it can not only affect proximal sites like intestine, but also have an effect on distant anatomical sites such as brain, lung, and joints. There are little studies about dysbiosis and factor VIII immunity, and one of the studies was actually done by a previous PhD student of our group, in which he had this group of mice who created dysbiosis by using broadest spectrum ampicillin to manipulate their gut microbiota. And interestingly, this study showed that mice which received ampicillin showed higher anti-factor VIII antibodies compared to control group. So there is a link between gut and liver through this gut-liver axis. Actually, liver receives 75% of its blood supply from intestine through portal vein, and in turn, the liver releases bile acids into the biliary tract. The endotoxins that are produced by gut microbiome can induce a pro-inflammatory liver microenvironment through the activation of Kupffer cells and subsequently inflammatory cytokines. Thus, alterations in the gut microenvironment are recognized to alter the inflammatory state of the liver. And again, in gene therapy, we are directing and targeting liver. So we propose that maybe these alteration in inflammatory state might have an effect in vector transduction, transgene expression, and vector persistence. So first, we needed to establish a model of dysbiosis. 6 mice were treated with an antibiotic cocktail containing ampicillin, neomycin, metronidazole, and vancomycin. It was administered through oral gavaging for 10 days. After 10 days, we had 3 endpoints, 2 weeks, 4 weeks, and 8 weeks. In each endpoint, 2 mice were euthanized, and the cecal content was collected. DNA was extracted from cecal content and then sent for 16S rRNA sequencing for microbiome analysis. Another 2 mice didn't receive this antibiotic regimen to count as control. Gut microbiome data was analyzed with CHIME2 pipeline. And in the heat map here, you can see bacterial composition between the control group and the antibiotic treated. Red boxes represent the abundance of that type of bacteria, and blue represents the lack of that bacterial group. And it is shown with this heat map that the control and antibiotic treated samples had different microbial composition. We also performed alpha diversity, which is a metric that measures the richness and diversity within a group of samples. And again, we can see that mice which received antibiotics show a reduction in diversity of bacterial composition compared to control group. Okay, let's take a break from Factor VIII and talk about alpha-1 antitrypsin. We will understand why in a second. Alpha-1 antitrypsin is a serum serine protease inhibitor, which functions to protect the lung from powerful neutrophil elastase. It is natively produced in the liver hepatocytes and then reaches the lung through circulation. In AAV gene therapies, we use human A1AT cDNA as a reporter transgene. And the reason that we do that is fourfold. First, human A1AT cDNA has a very small cDNA. It is 1.25 kb, so it can be easily packaged into AAV vector. It is not immunogenic in wild-type mice. There is species-specific ELISA to measure human A1AT, and using this reporter transgene will enable us to assess a transgenic protein that, in contrast to Factor VIII, is actually natively synthesized by hepatocytes. Okay, moving back to our project 2. So we established a model of dysbiosis by administration of antibiotic cocktails. Now we want to mix the antibiotic manipulation with the gene therapy. In here, we have 4 different cohorts of 4 different study cohorts. The first one is normal wild-type C57 black mice. And these mice underwent antibiotic treatment, again, for 10 days, 4 different antibiotics. At day 11, a majority of them received human alpha-1 antitrypsin AAV5 vector, and some of them were sacrificed to collect cecal content for microbiome analysis. Starting from 2 weeks post-vector delivery, we started our biweekly retroorbital sampling till 6 months. At 6 months, we had an endpoint in which we euthanized a group of these mice. And from 6 months to 8 months, we performed monthly RO sampling. And at 8 months, we terminated the study. We euthanized mice and collected blood and liver. Again, liver will be used for our studies in project 3. And the plasma samples that we collected during these 8 months will be used to measure human A1AT using ELISA. Another group of mice underwent the same timeline, except that they were not treated with antibiotic to count as a control. The third group of mice were our hemophilic mice, Factor VIII knockouts. They also underwent the same treatment and the same study timeline as this group. And the only difference is that they received K9 Factor VIII as a transgene. Another group was used in this for this transgene, which they did not underwent antibiotic treatment to count as a control. The plasma samples that were collected during the 8 months were used to measure Factor VIII activity levels using chromogenic assay. And again, the liver was collected to be used in project 3. So again, we confirmed that we manipulated their gut microbiome composition. These figures showed the microbiome analysis of wild-type mice which received human alpha-1 antitrypsin. Again, we can see a clear difference between the microbial composition and the fact that antibiotic-treated samples had reduced richness in bacterial groups compared to control. The same goes with samples that received. The same goes with the samples from Factor VIII knockout cohort. And again, those who received antibiotics showed reduced microbial diversity. So let's see what the transgene levels look like. First, starting with human A1AT transgene levels. we can see in the figure that starting from 1 month post-vector delivery, we can see human A1AT expression to about more than 20 nanogram per ml. It peaks in 3 months and then drops at 4 months. We still have samples that needs to be processed till 8 months, but for now we have the results till 4 months post-vector delivery. And we can see no significant difference between the pattern of transgene expression with antibiotic-treated and non-treated samples. This figure is actually the same as this, but the difference is that we can see variability of transgene expression in individual mouse plasma samples. Moving on to the canine factor VIII activity levels, our study groups that received antibiotic and those who left untreated with antibiotic. Starting with factor VIII knockouts that received antibiotics, we can see a factor VIII activity level starting 2 weeks post-vector delivery. It peaked at 1 month and then a sharp decline at 2 months to about 20% of activity, which remained stable for the remainder of the study. We have chromogenic results till 5 months post-vector delivery, and the other time points need to be processed. In control group, kind of the same pattern was seen. However, we can see a peak of increase in factor VIII activity levels 2 months post-vector delivery, and again, a decline at 3 months, which persisted for the remainder of the study. Again, this figure shows individual factor VIII activity levels. What it is not shown in these figures is that in here, when we see a sharp decline in antibiotic-treated samples, we actually lose factor VIII expression completely in 50% of the plasma samples that we have. And we thought that maybe it's an inhibitor reason behind this. We tested that. The data is not shown here, but we did a factor VIII binding assay and we confirmed that these samples had inhibitors. So the inhibitor resulted in this drastic fall in antibiotic-treated samples. So the main findings till now for the microbiome project is that liver-directed transgene expression was not influenced by the gut microbiome manipulation. So for sure, we know that vector transduction is not affected by microbiome manipulation. We can see a decline in transgene expression levels after a peak at 1 to 2 months of vector delivery, just what we see with preclinical and clinical researches. And in factor VIII cohort, 50% of antibiotic-treated and 28% of control samples developed inhibitors, leading to a total loss of factor VIII expression. So moving on to project 3. As I mentioned earlier, project 3 binds the results of project 1 and 2 and works on the samples that we received from project 1 and 2. In this project, we hypothesized that epigenetic modification to vector sequence contribute to the decline in transgene expression and induce production of factor VIII transgene protein in non-native cells, results in unfolded protein response, ER stress, and apoptosis of cells, which eventually result in the decline that we see in transgenic protein. So having the mouse liver samples collected from project 1 and 2, we will perform these 3 main experiments. First, we will quantify vector copy number and mRNA using qPCR and qRT-PCR to prove the decline in transgenic factor VIII plasma levels in liver tissues. Next, we will perform epigenetic profiling using ATAC-seq and cut and tag technology to evaluate repressive and permissive acetylation and methylation marks. And eventually we will measure GRP78 levels and caspase-3 as biomarkers of unfolded protein response and apoptosis of hepatocytes. So upon completion of these 3 projects, we will be able to answer these 3 questions. We will understand at which level silencing is enforced and what molecular characteristics lead to the silencing of the foreign vector DNA. Is the novel native enhancer that we use more resistant to epigenetic silencing? And how inflammatory liver microenvironments can influence AAV vector delivery? With that, I would like to end my talk and thank my supervisor, Dr. Lily Crabb, my supervisory committee members, Dr. Greer, Dr. James, and Dr. Hay for their scientific advices, the members of Lily Crabb Lab for their technical and emotional support, previous postdocs and PhD students, and CIHR Foundation, Biomarine Pharmaceutical, and Vanier Canada Graduate Scholarship for funding this project. Thank you all for your attention. I'm happy to take any questions.
Okay, questions, and if anybody has questions on the Zoom, you can put it in the chat or just use the hand up function and we'll keep an eye on you. Paula, go ahead.
The inhibitors.
What assay? Factor VIII binding assay.
As you don't know, like you haven't done the past yet.
Not yet.
Yeah.
And the reason is that I did this Factor VIII binding assay because I had limited amount of plasma samples from the RO sampling.
Okay.
And I haven't done Bethesda, which requires at least 50 microliter, of plasma samples, but it's on the to-do list to perform. Yep.
Is there any insight on project 1? With the enhancer, what are the transcription factors that bind there? And how come in the mouse experiment, whether you have the enhancer in the correct orientation or the reverse, it had roughly the same expression level?
So in silico analysis that we did within the enhancer regions showed transcription factor binding sites that were kind of ubiquitous. It was not liver specific, but like we also found like NF-kappa B, which is an inflammatory transcription factor, which also correlates with the fact that Factor VIII itself is an acute phase protein. So it has something to do with Factor VIII activity levels. And regarding your second question, I don't know if I get it correctly, but there is no previous research on employments of these enhancer elements in an AAV vector and introducing them to mouse models. So was that your question, or?
I was curious to see that when you had inverse sequence that the levels were roughly the same as
So we confirmed that these elements are enhancers by cloning them in both reverse forward and reverse orientation in plasmid, and we test that in vitro and in our in vivo cases. We will not be proceeding with generating AAV vectors that has both directions of enhancers because AAV generation is not as cheap as doing plasmid delivery. So we will continue with only forward enhancer elements. Yeah.
Monty first, and
All right,
Cindy?
just talking about the enhancer. So you took this C fragment, a fragment C, made a C prime.
C prime, yeah.
It strikes me that cutting out only that for enhanced expression so drastically when that same fragment is present in the full C. What is it saying about what else is present in the fragment C?
So I'm repeating the question for the Zoom
Yeah.
audience. I forgot for the
Thank you.
previous questions. So the question is, how come we see increase in luciferase expression with the shorter fragment, which is 400 base pair, but we do not see that with the fragment C, which is 900 base pairs. So this is one of the observations that kind of puzzled us because I expected to see increased expression at least at some to some similar levels with the shorter fragment. Once we saw that, I went and searched in like the UCSC genome browser to search for maybe repressive marks. We did not identify repressive marks doing in silico analysis, but with project 3, we will be able to maybe identify these repressive marks present in 500 base pair of the fragment C that maybe inhibits the expression.
Hi. Could you just talk a little bit, you were talking about how you're gonna take your novel C prime best show group, you know, when you're developing them and comparing them to transthyretin. Could you just talk a little bit more about why you think that your C prime derived enhancer elements are gonna be better than the current transthyretin?
Based on the in vitro and in vivo results that we got so far, we see this potential in this enhancer element to be able to increase expression of the downstream gene. Currently, the transthyretin enhancer is used in AAV gene therapy as a standard liver-specific enhancer element that enhances the expression of Factor VIII. Again, based on the results that we got, we propose that maybe these enhancer elements, which is derived from a native Factor VIII locus, will derive better expression compared to transthyretin, but we need to test that. Yep.
Have you done any plasmid expression studies with, you know, head-to-head without the AAV element?
Not yet, no. But that's a good point.
So picking up on, I think, where both Yvonne and maybe Monty were going, using this Jasper tool, you presumably get information about the cis-acting elements that are in the DNA, but also what the known transcription factors that are capable of binding there.
Mm-hmm.
So do those transcription factors line up with things that you would expect to be expressed to high levels in hepatocytes, I guess?
So yeah, as I mentioned before, the transcription factors that we could detect with a score of more than 400 in use in Jasper were ubiquitous. There were not where we were looking for transcription factor sites specific for endothelial cells, because Factor VIII is produced predominantly in liver sinusoidal endothelial cells. So I was looking for
Things that would give you
things like that, but it was basically ubiquitous TF binding sites, and the most relatable TF binding site that I could detect through Jasper was the NF-kappa B, which I explained.
Okay. So, but most of your data is based on hydrodynamic delivery, right?
And the in vitro.
And okay, that's right, in the cell line.
Yes.
Okay. All right, so you get a little bit of information, but not enough to figure out what deleting other sequences is really doing to help you.
Yeah, we are
Can
working
you
on that.
can you tell us a little bit more about AAV? What is it that adenovirus does for AAV?
So like I said, it's a helper virus. It helps in the replication of AAV.
How?
Well, it is a good question. I should have reviewed this information, the background. So I'm just trying to refresh my memory.
Because it's got a
already read
itty
that.
bitty genome.
It has a very small genome.
So-
It doesn't have all the required genomic elements for a full replication. So it requires, I'm just thinking what parts of it requires adenovirus, which my memory is not helping right now. Sorry.
Okay, that's fine. I mean, I don't remember either. And I wasn't aware that it was referred to as a dependo virus,
It's
is that
like a
relatively
dinosaur.
new name? Did you guys-
Dependo virus.
No,
David's been around.
I've got to check in my field virology textbook from the sixties and see if dependo viruses is a real.
And a part of parvoviride family. Like,
Okay. So the last question I had, other people have questions? In terms of, immune responses that's a big problem with Factor VIII therapy. Why do you think that AAV delivered, virus, transgenes like this are going to evade that, or will they?
well, in the AAV trafficking and AAV transduction that I explained, we can see immune responses against the capsid. So it will elicit adaptive immune response and the cytotoxic T cells, which results in eradication of transduced hepatocytes, and consequently,
So you got that problem, but do you think that you'd still get neutralizing antibodies against
Yes.
the Factor VIII that's produced
yes,
through this mechanism?
so
Okay.
CDLs and
Is
humoral
is that
immunity.
known from the trials that have been done so far with AAV,
Yeah,
if the neutralizing antibodies develop?
yes, yeah. I did not present those because there were a lot to present already. Yes, Tashika.
I was wondering regarding, in patients you mentioned in Factor VIII, gene eventually happening down. Is the gut microbiome altered in these patients? Is there some population where it doesn't affect? And does the gene therapy also affect the gut microbiome?
So the question was in the patients that received AAV5 in the beginning of my talk, did these patients undergone gut microbiome manipulation, and whether gut microbiome manipulation has an effect on AAV gene delivery, right?
If the gene delivery affects the gut.
Oh, if the gene delivery affects gut microbiome. So regarding your first question, no, these patients were not manipulated with their gut microbiome, and our study is actually the first study that investigates this relation, and whether AAV will affect gut microenvironment. So I'm just thinking about it right now, because if we say that upon AAV delivery, we can elicit immune responses and maybe inflammatory microenvironments of the liver, because it can happen through, and it is documented through measurements of alanine aminotransferase, or ALT. So it can affect the inflammatory states of the liver. And since liver and gut is also related, so maybe this inflammation can be transferred to gut microbiome and affect gut microbiome composition. So yeah, maybe. Good question. Thank you.
Any other questions, folks? If not, join me in thanking Fateme for our nice talk.
Thank you.